thank you all for coming today. I'm super excited about it. And I'm imagining by this point in time, you all have wonderful but pretty different ideas of what you would do with these data types that we've walked through today. And I think that's one of the coolest things about MONet. So what I'm going to show you today is our first results from a couple of brains, just looking at the 1,000 soils data set that we've produced. Hopefully it sparks your imagination and continues to show you different things that you can do with your data. We're really excited to start working with you guys, answering different questions, both at the local and the continental scale. So if you think about whatever your environmental issue of choice is, whether it's wildfires, hurricanes, ag, you name it, there's probably a substantial amount of uncertainty in both our understanding of those processes and our ability to predict that in the future. And so over the last couple of decades, there's been a lot of new research emerging in molecular science, both microbial, structural, and geochemical, kind of under the premise if we could just really understand what is happening in more detail, we would be able to predict these processes better and basically solve all our problems. So we know that's not happened yet, and we have a lot of work to do on that front. And that's what we're really hoping MONet can do. So when we think about soil carbon cycling in particular, which is what our pilot was based on, this research has really taken two avenues. One, I would say, focuses in on carbon chemistry. So this is the MEMS model from Francesca Controfo's group. And it's really honing in that substrate chemistry matters. And what they've done is they've divided our bulk carbon pools into several different pools that have different functional attributes and cycle different ways. And really the other avenue this research has taken in soil carbon cycling is microbial access is key. So hydrology, mineral associations. And so this is a nice paper by Bonnie Waring et al. That shows carbon flowing through the system, and really that microbial access can be a key component of its cycling. So MONet is broadly structured around these two premises. I'll walk through today our data from our 1,000 soils pilot, two examples of what we've done in it, just to show you the distribution of soil cores we collected in the pilot. It's pretty good across the continental United States, in large part to collaborations with NEON, some of our SFAs funded by the DOE and different academic partners. You can see at the bottom a range of soil parameters. We're covering a large range of not only ecoregions, but different geochemical environments as well. I'm going to focus here today, touch on some of our flagship measurements at EMSL, things that are a bit unique to our network versus other networks that are out there. One is poor network structure. I'm not going to go too deep into that. I'll let Tomas and others talk about that later today. But I will show an example of that. Organic matter chemistry, I'll focus on a little bit more. And importantly, how do we start to scale these variables and associate them with microbial respiration? So I don't pretend to have all the answers on that, but I will tell you some of the things we're starting to think about, and hopefully you can think about other ways to do that. So the two examples I'm going to give are at very different scales. The first one's at a local scale, led by Maggie Bowman. There's a paper out on it if you want more details. Really looking at the first 3 cores we received across a wildfire gradient. Looking at soil structure changes, respiration changes, and organic matter chemistry changes. And then Chen Shi, who's here in the audience, has looked at a continental scale, trying to employ some new machine learning approaches with Marie and others, looking at these continental scale processes and trying to understand what at a continental scale influences respiration. So you can talk to Chen, or he's got to talk at ADU that'll have more details on that. So first with Maggie's study, this is a really nice collaboration with the Warm Springs Reservation in Oregon and some partners at Washington State University. We took 3 cores across a burn history gradient. So we have 1 core WSU3 that is unburned and 2 that have different levels of previous burn history. And so this is the same study that Will showed a slide on earlier, but I'll give you a little bit more detail on it. And so what Maggie did was, was first kind of look at soil structure differences. And if you just look here at color and physical differerences, you can pick out by eye, right? The most burn core is darker and perhaps more condensed than the unburned soil. And so she, with Tomas, used X-ray computed tomography to quantify these differences. And what she found is really in the surface layer is where we see the biggest effects. The burning really decreases pore connectivity and porosity in the surface, where more insulated layers at the bottom don't have as large of structural differences. So then her interest was really in the organic matter chemistry side. I won't go into this in as much detail because Malak and Will did a really nice job on it. Just to say that you can get a ton of information out of FTICR data. And this is really an emerging field, how much information we can get out of it. And so Maggie primarily looked at these different carbon classes and then our nominal oxidation state of carbon, which Will talked about in a little bit of detail. The important thing to recognize here in terms of NOSC is that our more positive values are more oxidized. So this is a nice paper by Marco Kilowait. And as we move from lipid-like compounds all the way up to proteins and nucleic acids, we get more and more oxidized carbon molecules. And that's reflected in this NOSC value. So I know this figure is a lot to look at. I'll kind of walk you through it. So these are distributions of NOSC on the x-axis, density distributions. So the solid lines are the top layer of the core, the dotted lines are the bottom layer of the core. And if you just look at where these arrows are pointing, they're pointing at roughly the means or the medians of each type of group. And so you can see in the surface layer, if you just look yellow to dark red, so unburned to more burn history, we're seeing an increasing carbon oxidation with fire history. That's exactly what we would expect to see because combustion generates more oxidized organic matter. So this is a really nice linear increase in the surface layer. And the bottom, we see a similar trend from unburned to burn, getting more oxidized. But really in this layer, there's not a linear increase. So it matters, is it burned or not burned? And I think this is really interesting because that bottom layer is more insulated. And one of the pressing questions in wildfire science is how far do those effects go down? And that varies with a lot of things like fire intensity and frequency. She also looked at just compositional differences. So this is grouping all depths together, looking at burned versus unburned. Again, you see this binary effects where just having previous fire history changes organic matter profiles substantially. Malak did a really nice job of introducing where these things came from. If we look at our burned soils and we do some of the things Malak was talking about, so our transformations and our mapping to microbial metabolisms, we can start to pull out processes that are associated with burned versus unburned soils. And again, we see things we might expect in our burned soils. So polycitric aromatic hydrocarbons, or PAHs, are known combustion products. They're really large contaminant and they're persistent versus degradation is a key issue in environmental science right now. So we see that microbial metabolism going on in the burn soils. We also see, when we look at transformation, carboxyl groups being transferred back and forth, which is, again, we would associate with combustion byproducts. And what's really cool about these, this is only 3 cores. So we don't have robust statistics to actually do an analysis here. And that's kind of cut off. This burning is associated with lower rates of microbial respiration and lower microbial biomass in those burned soils as well. So we're starting to be able to link physical changes in soil properties, organic carbon changes with changes in microbial biomass and respiration. And so this was our first study out. It's really exciting in terms of the potential that you could do at the local level at your study sites with MONet data. If you want to know more about this, Maggie's got a really nice paper out that we'll show as well, so you can look at that in more detail. So now we're going to go to the entire opposite end of the spectrum. For those of you who might want to do synthesis or meta-analysis type investigations. So Chen has used some cool new machine learning approaches to try to decipher patterns out of these really complex data types. So I'll show you a little bit about his workflow. You can talk to him for more detail. The first thing he did, focusing just here, this is only going to show organic matter chemistry, biogeochemistry, and respiration, no structural analysis in this study. So he took his FTICR data and tried to distill this information down into a subset of features that described our carbon pools. And for that, he used something called non-negative matrix factorization. Conceptually, you can think about it similar to principal component analysis. I'll show you why that analogy is important in a little bit. But if you know nothing about NNF, think about it for now as a PCA. Then he looked at how well these different components could predict respiration, especially in comparison to biogeochemical variables like total carbon, soil moisture, things that are commonly measured. And then he did some model evaluation and looked at compositional differences when this data was summarized by NMF. So really the first exciting outcome is that NMF is summarizing data into tractable units. This is a lot to look at. I'll walk you through how it's structured. So each column here is an NMF. So you can think about that as a principal component, basically a vector that summarizes carbon chemistry into discrete units. Here on the left-hand side, so this bar with different colors is just the chemical class. So Chen has clustered them by what type of molecule they are. The big bar is lignin, and then you can see tannin, all the way down different types of chemistries. And then what's on the heat map is specific organic molecules going from blue to red, different weightings of how important that molecule is for the different component. And what you really need to see, if none of that made any sense to you, is that the red patterns are very different across these 7 different components. And so we're getting separation across different chemistries, down from tens of thousands of compounds here in the surface soil to just 7 different vectors we can now work with. Similar thing in our subsoil. So these are greater than 10 centimeters. We get really nice separation. The model here only has 5 components. We can talk about why that is in a different venue or afterwards. But you can see it again, particularly like NMF5 and 2 have really strong signatures in different chemical portions of the spectrum. Relating this to soil respiration, what I want to show here, so these are different groups of soil respiration, low, medium, and high. The stack bar plot, this is surface soils. So the colors here are seven different NMFs. If you look from low to high, you will notice that there's very different stackings of the NMFs in low versus high respiration. And so like this means that NMF 6 is not found at all in low respiration samples where it's really high in the high respirations. And so when we relate that back to the chemistry that we saw in the previous heat maps, excuse me, we're seeing things that make a lot of sense ecologically. And so this green NMF, NMF 3, that decreases from low to high, when we look at the chemistry of that, that is mainly lignin and tannin. We would expect that to have low bioavailability. I get really stoked about NMF 4, which is only present in low respiration samples. That's protein and lipid-like compounds. So we're thinking maybe that's microbial necromass, which we know to be really stabilized. And then if we look at the high respiration samples, we're seeing a super large diversity of compounds that are associated with high respiration, notably amino sugars that are thought to be really bioavailable. Just to show you what the bottom looks like, it's a similar story here. If you dive into the chemistry and low respiration, we have a lot of lignin and tannin, and then we have high diversity, including amino sugars in the high respiration samples. So one thing he wanted to do was, okay, we have these really nice summaries of organic matter. We can break them up by respiration groups, but can we really assess to what extent these help us explain respiration? And how does that compare to things that we typically measure? And so what he's done here, I'm just showing surface soils here for a reason. He's built a regression model that uses just our various biogeochemical variables that we measure, tries to explain respiration, explains 44% of variation, which when you think about the different soils we measured at the continental scale, I actually think that's pretty good. But what was really cool is if we just do no biogeochemistry and focus it on our soil organic matter patterns, we actually get about 10% more variation explained in respiration rate just by using the water extractable organic matter. And then when we put them together, we get even higher explanatory power. So you might notice that if you can see, the top variables here are still biogeochemical variables, which makes sense. It's sort of the traditional paradigm, but we are getting a lot of added predictive power from 44 to 62% by adding these metrics of carbon chemistry in this model as well. One thing I was really interested in, if we look at the bottom soils, these models do not work very well for soil organic matter chemistry. So we still get decent biogeochemical variables, but we don't get much predictive power of respiration from soil organic matter chemistry. We think that's probably due to mineral associations in that layer. And so one of the things MONet has done is actually start measuring mineral associated organic matter as well. Here's our conclusions from that. Basically, these machine learning methods seem to do well and produce like ecologically relevant results that make sense. Our top and bottom layers are very different in composition, which you might expect. And in that surface layer is where we're getting a lot of added value for predicting respiration from our dissolved SOM pool. If you want to know more, Chen's talk at AGU is on Monday for those of you that will be there. He's also here hiding if you want to talk to him later about that. This is just to bring it back that these are two examples. There's a ton of different things we can do with this data. And you guys, I'm sure, all have such a diversity of ideas. You're probably all thinking something different. We have a lot going on our side to help facilitate this, but this is really about collective science, both at your local study sites and at the continental scale, helping us understand how carbon cycling works, how it might impact our climate, responses to disturbances, so all types of environmental issues. And there's, as you've noticed, there's a whole team of us in the Army working on this. So depending on what you're interested in, we can hook you up with one of these folks and get more help on that. Cool.
Emily, we have time for questions from the audience or from here in person. You might come over there too. Okay. But I actually, Emily, I was curious not to put more work on right now, but how quickly can those NMF, plots to be generated? Because I'm very curious about your burn sites and if that will show any difference in those sites as well.
So in my understanding now that we've worked out kind of the workflow and shown that it's producing good results, it's a fairly quick workflow. Rudy has helped a lot with that. In my opinion, the biggest thing about machine learning is anybody can build a model, but you want to make sure it's good, right? Like being a good fitting model and all that sort of stuff. We don't right now have a workbook or example code to do it, but that's definitely something we're interested in. And that should be a pretty quick thing once we get that developed. Well, yep, Marian.
And actually sticking with the NMFs, if you have like different samples, how reproducible are the groupings? And I'll give you an insight into why I'm asking. It reminds me of the Parafac analysis done for fluorescence dissolved organic matter. And one of the strengths of that was that once people put all their data together, they found like common groupings and they standardized the nomenclature of those and they can relate them across studies. And it made a really cool data set because they had like a training data set. You trained your FDOM, your EAMS based on this sort of global database.
Yeah, so I think that's a really interesting question. It is more like principal component than Parafac. So what you put in will influence how it all groups or clusters together, right? So you won't be able to take like NMF3 and run a different data set and have NMF3. However, I would hope that the specific compounds that are coming out that are important for respiration would be somewhat consistent across different sample sets. So that's the hope, that we can use this sort of approach on any data set and start to pull out the things that are important. Amir.
Yeah, thanks for the great talk, Millie. I have a question about the wildfire experiments and data. So it seems like you have different sites, burned and unburned, and you see some differences with different data that you collected. I'm just wondering how we can make sure that the differences that you see is exclusively because of the wildfire and burn and unburned. What about the other variations, like vegetations, like microbial communities? I mean, have you considered those other variations as well, or you can definitely say that these differences are just because of wildfire?
That's a great point. So that's a challenge with all environmental work. So in this study, the results make sense in the context of wildfire, but we can't exclude anything that you mentioned. I will say that we are, as we get more and more samples, we're getting more and more wildfire, we're getting, more and more ag. So the hope is that, if you're a wildfire person or an urban person or whatever, you can take not just your study site, but you can take, 50 wildfire sites across the country that have different vegetation and different properties and start to make comparisons where you can kind of isolate wildfire effect there.
just quickly following up on that, Emily, then how much of that is planning to be ingested into MONet? Like when someone submits a core, is that level of metadata captured with it? Or only the people who have submitted the cores kind of know that information?
Yeah, so that disturbance type is a metadata parameter that we are collecting. So that should be searchable in our database. Disturbance type, or if they did any type of amendment, anything like that, that should be metadata that you will eventually be able to search on.